Hello student, today we are going to see about DNA sequencing. So DNA sequencing means that is determination of nucleotide or base sequence of DNA, where we are determining the ATGC sequence of DNA. For the DNA sequencing, we have several different methods like Maxam-Gilbert sequencing, Sanger sequencing, automated DNA sequencing, pyro sequencing, Roche 454, solid sequencing, Illumina sequencing. So these are the different methods we are using for the DNA sequencing. Okay, the Roche, solid and Illumina, these are called next generation sequencing methods. So today we are going to introduce about the Roche 454 sequencing. Okay, so Roche 454 is a one type of NGS sequencing method. Okay, the basic principle behind the Roche 454 is involving the amplification of DNA fragment, which is captured on the microbed as a reactor. Okay, using immersion PCR to generate large enough copies of fragments for the sequencing, followed by sequencing of amplified fragment by pyro sequencing. Okay, so in this sequencing technology, the DNA, they are firstly fragmented. Okay, and then this fragmented DNA, they are captured on microbed, where the immersion PCR is carried out for this captured DNA to generate large number of copies of this fragment. And after the generation of large number of copies on captured microbed, the DNA is sequenced by using pyro sequencing technology, that is sequencing by synthesis using 4 different enzymes. So this is the basic principle behind the Roche 454 sequencing. The basic step involved in the Roche 454 sequencing, that is first the sample preparation, which involves the preparation of DNA samples, then loading of these DNA samples on the microbeds, then carrying out the immersion PCR, and finally the sequencing by using pyro sequencing by synthesis method. So these are the basic steps here, where you can see the basic steps for the Roche 454 sequencing method. These are the microbed on which we are loading our DNA fragment. Okay, and then they are amplified in emulsion PCR to generate large number of copies. So we will going to introduce each one of these steps in detail. So first step in the pyro sequence 454 Roche sequencing is the sample preparation. Okay, the DNA double helix DNA strand ladder or DNA template strand which we want to sequence is firstly cut into small double-stranded fragment of approximately size 400 to 600 base pair. Okay, so double-stranded DNA is firstly made, fragmented into approximate 400 to 600 base pair fragment size. Okay, and after that this generated fragment, they are made blunt-ended using the appropriate modification enzymes. Okay, which modify the ends of DNA and thereby makes it the blunt-ended. After the DNA fragment they have made blunt-ended, we are attaching two different adapters, A adapter and B adapter, to this DNA fragment. Okay, at two different ends. The A and B adapter, they are used here as a priming site because the sequence of this A and B adapter we already know. So their sequence is used to design the primer or primer binding site, both for the amplification as well as sequencing primer. Okay, so A and B adapter, they help us as a priming site for the both amplification as well as sequencing. Once we have attached the adapter to the DNA fragments, the fragments, they are made single stranded using a treatment either denaturation by heat or sodium hydroxide. So double stranded DNA fragments, they are then made into single stranded using denaturation treatment. Okay, either heat or use of sodium hydroxide. So this is the basic step of sample preparation, which involves fragmenting the DNA, then making it blunt-ended, then attaching A and B adapter as the priming site for the amplification and sequencing to both ends of the DNA. Here you can see the adapter, they are attached, A adapter at one end and B adapter at other end of the fragment, which are single-stranded after the denaturation. After the adapter, they have ligated and the fragment, they are made single-stranded. This adapter ligated fragments, they are then attached on the micro beds. Okay, So next step in the 454 Roche sequencing, that is loading this DNA sample onto microbed. The DNA library fragment, they are put on the micro-size bed by mixing together, okay, and this fragment will attach on the microbed because they contain adapter B, which has tag that is called 5' biotin tag. This biotin tag helps for the immobilization of this fragment on the micro-size bed, okay. This micro-size bed, they are magnetized, that is they have attached streptavidin, they are coated with streptavidin, okay. This streptavidin has strong affinity towards the biotin, and therefore the adapter B, it will go and bind to this beds. So beds, they are firstly made magnetized by attachment or coating with streptavidin, and thereby it will attract the biotin in the B adapter and thereby help the attachment of this adapter ligated fragment on the micro-size bed. Okay, so this step is called loading of the DNA sample onto the bed. Okay, after the beds, they are loaded with DNA fragment. Okay, this DNA library fragment along with capture bed, they are then subjected for emulsion PCR, and for this, these beds along with PCR reagent water, they are mixed into the small cylindrical plastic containing synthetic oil, where vigorous shaking of this PCR water mixture and the oil mixture lead to the formation of a small droplet around the bed, that is called emulsion. Okay, so vigorous shaking of the both this mixed component will lead to the formation of emulsion, that is a small droplet around the bed containing DNA fragment, which include each one droplet contain one DNA fragment and all PCR components which are mixed in the water. Okay, so each emulsion contain one DNA fragment which is captured on the micro-size bed beds and containing the water mixture of PCR components. After this formation of emulsion, the isolated DNA fragment in each droplet will be get amplified into the millions of copies by PCR reaction that is carried out in a each droplet, that is called emulsion. Okay, so each DNA fragment which is captured on the bed is going to multiply into millions of copies of DNA fragment. Okay, after that after the emulsion PCR, the beds, they are screened to eliminate only beds which are not containing any DNA fragment and the beds which are containing more than one type of DNA fragment. So we will enrich our beds only for those DNA which are only single type and they have attachment of single type of beds. So screening of bed help us to enrich the micro beds containing only single type of DNA fragment. So here you can see how this is performed. Once you have adapter ligated, the adapter, they are attached with the micro-size bed, and after that this micro-size micro beds attached with adapter, they are mixed with the tube containing emulsion oil, a synthetic oil, along with PCR mixture in water, and therefore vigorous shaking of this both component will lead to the formation of oil droplet around the microbed, that is called as a microreactor, because each microreactor contain one bed attached with one type of DNA fragment and water mixture containing PCR compound. So here the DNA fragment which is attached to this micro size bed will be get amplified into the number of copies and will be remain attached to the bed. Okay, so emulsion PCR will carry out the formation of multiple copies of DNA fragment which is attached to the micro size bed. After that, the micro size beds, they are break down and isolate the DNA containing the beds. Next, this step in the 454 sequencing, that is sequencing, and the sequencing mainly is based on the pyrosequencing technology, where sequencing by synthesis method is carried out to sequence the DNA fragment. Here the DNA capture beds, they are loaded firstly on the micro titer plates. Okay, so whatever DNA capture bed, they containing multiple DNA copies of single type, DNA fragment will be then loaded on a picotiter plate which contain about 1.6 million swings for the DNA sequence. So after the loading of this micro-sized bed containing the multiple DNA copies of single DNA fragment, they will be loaded in the picotiter plate, and after that we are loading the two another bed, that is one enzyme bed and the second packing bed, into the same well which contain DNA capture microarray. The enzyme beds, it contains the enzymes for the sequencing, that is DNA polymerase, ATP sulfurylase, okay, as well as it will contain luciferase, okay. So these enzymes, they are present in the enzyme bed, and the packaging bed, they are used only to keep the DNA bed in place within the well. So they help to maintain the beds properly within the beds, okay. So two beds, they are loaded after the loading of the micro-sized bed on the picotiter wells. The above well, this picotiter well, above this picotiter well, there is a flow channel, okay, which pass the nucleotide sequentially according to the schedule and a pyrase in a timely schedule, okay. So sequence one nucleotide at a time will be added according to the schedule within this picotiter plate wells, okay. So flow channel has a role to pass the nucleotide and a pyrase enzyme in a timely schedule. Okay. The sequencing in the picotiter well is based on the sequencing by synthesis, okay, where the bases, they are flown timely in a sequential manner in the same order, okay, during the sequencing run. And whenever the nucleotide complementary to the template strand, it will get attached on the template strand, it will generate visible light, okay, as we found in the pyrosequencing. And this visible light is then recorded by charge coupled device camera, CCD camera, and the strength of this light is directly proportional to the number of nucleotide incorporated. So here you can see the primer which is annealed to the nucleotide annealed to this DNA fragment. According to the primer is designed according to the one of the adapters that is attached to this DNA fragment. So after once we have added the sequentially the nucleotide bases, suppose this template DNA has a T nucleotide here. So when we have added the A nucleotide in the sequential run, the A nucleotide will get attached here and it will release a pyrophosphate. This released pyrophosphate is utilized by the enzyme ATP sulfurylase to form the ATP, and followed by this ATP is further used by the next enzyme in the sequencing of pyrosequencing technology, that is luciferase, which we will use this ATP to convert luciferin into oxyluciferin with the emission of light, which is proportional to the number of nucleotides. If there are two sequential nucleotides get added, then the more light is generated compared to the single nucleotide addition, okay. Based on this, we are determining the sequence of DNA. So here this is only a single strand shown, but here simultaneously we can run the sequencing on multiple DNA fragments of single type which is attached on single microbed. Therefore sequencing can be completed within a desired period. So here you can see the basic of pyrosequencing. When nucleotides, they are sequentially added within the reaction of pyrosequencing technology during the 454 Roche. Whenever the nucleotide will be found complementary and attached during the synthesis, the DNA polymerase will release the terminal 2 pyrophosphate, okay, and this terminal 2 pyrophosphate, they are used by the ATP sulfurylase to convert adenosine monophosphate into the ATP, okay. After that, this ATP, they are utilized by the enzyme luciferase, the next enzyme in the pyrosequencing technology, to convert luciferin into oxyluciferin with emission of light, which is proportional to the amount of nucleotide. If single nucleotide is added, the light generated is low, okay. If there are two consecutive same nucleotides are added, the amount of light generated is more. So based on the light intensity, the number of nucleotides added during the DNA sequencing can be determined. If any nucleotide is not get added during the uh, due to non-complementary. Then such nucleotide, for example here, the first nucleotide that is we have added in the sequential addition is adenosine triphosphate, but it does not found the complementary here, so it will be get degraded by a pyrase enzyme action into adenosine monophosphate. And like this, the sequencing in the pyrosequencing is carried out simultaneously on multiple DNA fragments, which is attached on a single microbase. Okay, and all these multiple DNA fragments, they are of single type. So like this pyrosequence, this one 454 Roche, it will carry out the sequencing based on the technology of synthesis by sequencing of pyrosequencing. I hope you have understood very well about the 454 Roche sequencing. Overall, 454 Roche sequencing involves sample preparation, that is loading the sample on the microscopy bed, and then carry out emulsion PCR and then sequencing by synthesis using the pyrosequencing technology. Thank you.